Welcome to the video class on cell cycle. Where a cell arises, there must be a previous cell. Just as animals can only arise from animals and plants from plants. This cell doctrine, proposed by the German pathologist Rudolf Virchow in 1858, carried with it a profound message for the continuity of life. Cells are generated from cells, and the only way to make more cells is by the division of those that already exist, and this remains as the basic function of every cell. So the basic function of the cell is to grow and divide. A cell reproduces by carrying out an orderly sequence of events in which it duplicates its contents and then divides into two. This cycle of duplication and division, known as the cell cycle, is the essential mechanism by which all living things reproduce. Cells on the path to cell division proceed through a series of precisely timed and carefully regulated events, namely stages of growth, DNA replication, cell division, and cell differentiation. To understand the cell cycle, let us know the phases or the stages the cell passes through in a systematic regulated manner. Cell cycle can be divided into two. They are the interphase and the M phase. Interphase in turn is comprised of three subphases, namely gap phase 1 or G1 phase, synthesis phase or S phase, and gap phase 2 or G2 phase. M phase is divided into mitosis, which is otherwise called as karyokinesis, where the division of nucleus takes place, and cytokinesis, which means the division of cytoplasm. Mitosis is further divided into prophase, metaphase, anaphase, and telophase. The pictorial presentation given on the top is a snapshot of the cell's journey. The division of a hypothetical eukaryotic cell with two chromosomes is shown to illustrate how each cell cycle produces two genetically identical daughter cells, and each daughter cell can divide again by going through another cell cycle. Before entering into the events occurring in various subphases, let us learn in brief about the interphase. The period between one M phase and the next is called interphase. Under the microscope, it appears deceptively as an uneventful interlude during which the cell simply increases in size. However, it is a very busy time for the cell, and it encompasses the three phases of the cell cycle. During interphase, the chromosomes are decondensed and distributed throughout the nucleus, so the nucleus appears morphologically uniform. At the molecular level, interphase is the time during which both cell growth and DNA replication occur in an orderly manner in preparation for cell division. During all of interphase, a cell generally continues to transcribe genes, synthesize proteins, grow in mass. Together, G1 and G2 phases provide additional time for the cell to grow and duplicate its cytoplasmic organelles. Looking into the events occurring in the G1 phase, the G1 phase is also known as the first growth phase or post-mitotic gap phase. In the G1 phase, the cell is metabolically active with rapid protein synthesis and growth. This phase may vary widely in its length. Cells that divide rapidly may do so in minutes to hours, but cells that divide slowly may last for days to years. Hence, the G1 phase is also called as variable phase in concern to time. To learn a few, a single-celled yeast can divide every 2 hours, whereas a mammalian liver cell divides on average less than once a year. It is characterized by a change in chromosome from condensed state to more extended state and series of metabolic events that leads to initiation of DNA replication. During G1 phase, chromatin fibers become slender, less coiled and fully extended and more active for transcription. The transcription results in synthesis of RNAs, tRNA, mRNA and rRNA and series of protein molecules required for initiation of DNA replication. All cell organelles like endoplasmic reticulum, mitochondria, Golgi complex, ribosomes and plasmid in case of plants multiply. G1 phase is very significant phase of cell cycle as the cell grows, increases in size and accumulates the building blocks of chromosomal DNA and the associated proteins as well as sufficient energy reserves to complete the task of replicating each chromosome. Initiation of DNA synthesis marks the beginning of the S phase, which is about 6 to 8 hours in duration. DNA is replicated so that identical copies of genetic material are received by the two future cells. All of the chromosomes are replicated. Following replication, each chromosome now consists of two sister chromatids. Thus, the amount of DNA in the cell has effectively doubled even though the ploidy or chromosome count of the cell remains at 2n. Chromosomes double their number of chromatids post-replication, but the nuclei remains diploid as the number of centromeres and chromosomes remain unchanged. Hence, the number of chromosomes in the nucleus, which determines the ploidy, remains unchanged from the beginning to the end of the S phase. The centrosome is duplicated during the S phase. The two centrosomes will give rise to the mitotic spindle, the apparatus that orchestrates the movement of chromosomes during mitosis. At the center of each animal cell, the centrosome are associated with a pair of rod-like objects, the centrioles, which are at right angles to each other. Centrioles help organize cell division. Centrioles are not present in the centrosomes of other eukaryotic species such as plants and most fungi. The cell now enters into G2 phase. G2 is also called second growth phase or pre-mitotic gap phase. In this phase, synthesis of DNA stops and synthesis of RNAs and proteins continues. All cell organelles multiply and spindle formation takes place. It lasts for 2 to 5 hours in most cells and may be as short as 1 hour in rapidly dividing cells or of nearly indefinite duration in some polyploid cells and primary oocyte that are arrested in G2 for extended periods. The first visible sign that a cell is about to enter M phase is the progressive condensation of its chromosomes. As condensation proceeds, the replicated chromosomes first become visible in the light microscope as long threads which gradually get shorter and thicker. This condensation makes the chromosomes less likely to get entangled so that they are easier to segregate to the two forming daughter cells during mitosis. Apart from regular normal cell cycle, the cell may decide or choose to enter into another phase which is called as the G0 phase or the resting phase. The G0 phase is often referred to as the resting phase and it is the phase in the cell in which the cell is neither dividing nor preparing for division. Hence, it is in a resting phase. And what really happens in this G0 phase? Or is G0 phase common for all cell types? Why at all a cell might enter a G0 phase? Or how can a cell exit from a G0 phase? What is the significance of a cell entering into a G0 phase? To answer these questions, let us consider not all cells adhere to the classic cell cycle pattern in which a newly formed daughter cell immediately enters the preparatory phases of interphase and follow mitotic phase. There are certain cells which don't prepare for division and remain inactive by entering the G0 phase. Sometimes it is understood that a cell exits from the cell cycle and enter the G0 phase by taking up this alternate route. The cell can either become a quiescent cell or a differentiated cell. The difference between these two cells is that the differentiated cell cannot get back to the normal cell cycle as the process becomes irreversible, whereas a quiescent state is a reversible process waiting for a signal. The two conditions where a cell decides to exit a cell cycle and enter the G0 phase is Number 1. Influence of internal factor, that is the genetic program allowing the cell to enter G0 phase, or any external factors like limited availability of nutrients and growth factors. As we all know that the cell division is an expensive process, the cell has to accumulate more raw materials and duplicate its cellular contents at the expense of ATP, which is derived from nutrients. Deprived quantity of nutrients makes the cell to think and end up with a wise decision of entering the temporary G0 phase, waiting for a favorable condition. Interestingly, a cell in quiescent state is very different from other cells in aspects of gene expression, metabolic pathways, and chromosome architecture. In fact, a cell spending its time in G0 phase doesn't mean it is in an inactive state. Rather, they are much functioning and active. For example, the nerve cells of our brain, where they remain electrically active in conducting nerve impulses and communicating with each other. The significance of entering into the G0 phase could be explained by considering the example of nerve cell, where it happens to balance between self-proliferation and differentiation into mature neurons. Talking about the regulation of the cell cycle in terms of checkpoints. Events in the cell cycle are ensured by a complex network of regulatory proteins known as the cell cycle control system. The cell cycle control system achieves all this by means of molecular brakes that can stop the cell cycle at various checkpoints. Some of the important checkpoints to be discussed here are the G1 checkpoint at the G1S transition, the G2 checkpoint at the G2M transition, the spindle checkpoint at the transition from metaphase to anaphase. The G1 checkpoint is the main decision point for a cell, that is the primary point at which it must choose whether or not to divide. Once the cell passes the G1 checkpoint and enters S phase, it becomes irreversibly committed to division. Hence, the cell checks for cell size, availability of nutrients, energy reservoir, growth factors, and any DNA damage. The G2 checkpoint checks for completeness of DNA replication and any DNA copy error. If errors or damages are detected, the cell will pass a G2 checkpoint to allow for repairs. If the checkpoint mechanisms detect problem with the DNA, the cell cycle is halted and the cell attempts to either complete DNA replication or repair the damaged DNA. If the damage is irreparable, the cell may undergo apoptosis or programmed cell death. Spindle checkpoint occurs during cell transition from metaphase to anaphase, where it is a check looking for the attachment of chromosome to the spindle at the metaphase plate in order to achieve separation of chromatids during anaphase. The next is the M phase. M phase includes mitosis, which is the nuclear division, and it is the most dramatic stage of the cell cycle, corresponding to the separation of daughter chromosomes, followed by cytokinesis, which is the cellular division resulting in two daughter cells. The descriptive explanation can be seen in the next video. Thank you all for listening.